All right, so this is exam 2, essay 2. I'm going to be talking about taste, smell, vision, and then hearing. And so just the 4 senses that we've already gone over. So if I were to, I'm going to start with taste and draw a very simplified tongue. So we can see. So this is the surface of your tongue. These are like little taste buds. So the little like papillae guys on the surface of your tongue. And then we have cells right here. They connect, they're over here too. And these are called taste receptor cells. So these guys are what actually transmit the information from the chemicals you taste on your tongue to your brain, and that action potential then turns into information. So it's very important that we have these guys. And so it's not our actual taste buds that taste things, it's these taste receptor cells that actually taste things. However, if we were to zoom in on these guys, it would look like a little box. And that box is going to change based on what you're tasting. So the basic box is going to be, I hope you can see that high. You're going to have the sarcoplasmic reticulum, sarcoplasmic endoplasmic reticulum. You're going to have a couple of vesicles. You're going to have a calcium link channel, and then you're going to have a voltage-gated sodium channel. And so this is then connected to a nerve, which then, when it interacts with that neurotransmitter, it's going to create an action potential, which allows you to taste. And so there are 5 different things we can taste. There's salty, and that's caused by things like sodium and potassium. And then there's sour, and then those things are caused by acids or hydrogen atoms. And then next we have sweet, and then these are caused by like glycerin or like, I'm just gonna write down sugar. Then we have umami, which is caused by glutamate, our good friend glutamate. And then we also have bitter, and then bitter is caused by calcium, or what we call quinone, and I'm just going to abbreviate it as Q. So these are the different substances that create these different tastes. And so these 2 are going to be different, and then these 3 guys are kind of going to work the same, depending on what type of substance it interacts with. And so we can start with salty. So I'm basically just gonna keep editing this box. This is just the basic, like the skeleton of what I'm gonna talk about. Each of them are gonna be different based on how they taste, kind of, whatever. So salt, you start out with your little box. But then you have something called an ENAC channel, which is an epithelial sodium channel. So this guy is open and allows sodium to enter into the cell. This guy has a sarcoplasmic reticulum, has these vesicles, has these channels. And then this entrance of positively charged ions is going to create an action potential, which then results in stimulation, and then you taste it. You taste something. That is how salt works. However, for there was salty, there was sour. Okay, so the next one is sour. So this sour one is also going to use the ENAC channel. However, it's not going to be moving sodium, it's going to be moving hydrogen ions, but it's just going to go through the same channel. And then this guy blocks the potassium leakage channel, which then increases positivity inside the cell, which then creates action potential. And then this guy can create stimulation, and you taste sour things. The next one encompasses all, like the sweet, the bitter, and the umami all together. They all kind of work similarly. So what happens is, instead of the ENAC channel, we're actually going to have a G protein right here. which is activated by a ligand, binds there. And then, I hope you can hear me well also, but a ligand binds to the G protein. This then creates more IP3, which then causes the sarcoplasmic endoplasmic reticulum, or the SR. It releases calcium, which then creates an action potential, and then you taste those things. So that's how you taste different things, by using these little taste receptor cells. So that is how taste works. All right, I put my headphones in so you can probably hear me a little bit better. I was just facing the board a lot while I was drawing. So the next thing we're going to talk about is how you smell. Smell is pretty simple, actually. If we have your nose, your mouth, and like the nasal cavity, your olfactory plate is going to be right here at the top of your nasal cavity. And then this guy, we're just going to zoom into him. So we have, well, I'll draw that lower so you can see it. Box here. It has all these globular cells that release mucus, because in order for you to smell something, it needs to diffuse into mucus, so then you can like smell it. But inside this is going to be all these little olfactory nerves, and these guys are going to be intertwined all up in this olfactory plate. So this guy then connects to your olfactory bulb in your brain, and then you smell things. So how you perceive anything is just stimulation, and then action potentials take it to the brain. However, usually things go to the thalamus first, and your thalamus basically filters everything out. But olfactory nerves don't go through the thalamus, and so that's why memories can be very strongly associated with smells, because you don't filter it out really. And then on these nerves, that's your skull. And so in order to get to your brain, these nerves need to go through your skull. I can't remember what this is called. It has a name, but has this. That's how you smell. If we zoom in on one of these guys, yeah, whatever. This is the end of it. It's gonna have another G protein right here. This is called adenylocyclase, something like that. But this is going to create CAMP, which is then going to open calcium and sodium channels on that nerve, and then create an action potential. And so this guy is just what receives the information on the mucous membrane. So that's how you smell. That one's also pretty simple and very straightforward. So next we get to go to vision. Woohoo! Everybody loves vision. Except me. I'm joking. Vision is actually super interesting. All right, vision. I'm gonna simplify this, and not go through exactly everything we went over in lecture. Well, maybe I will. I don't know. We'll see. We'll see how it goes. I'm gonna draw an eye really quick. This is an eye. You have a lens. You have your ciliary ligaments, your ciliary muscles, and then you have the pigmented epithelium back here. Light comes through here and hits the back of your eye. That's how you see. What is on the back of your eye, or your retina, is going to be a rod or cone, a bipolar cell, a ganglion cell. The ganglion cell is going to leave the eye on the, oh my gosh, what is this called? Is this optic disc? Optic disc. And then what's leaving the eye is the optic nerve. And so on this area of your eye, you don't have any photoreceptors, so you can't, that's your blind spot, when people refer to blind spot. This is what they're talking about. That's where all the ganglion cells meet up and move their action potentials to the brain. Because like I said earlier, the only way to perceive something is for an action potential to make it to the brain. So That's how it does it. I'm gonna zoom in on these three guys over there. But I'm gonna erase this first because I wanna draw big. So just keep in mind, this is what I'm zooming in on. It's at the back of the eye. It's your photoreceptor, your bipolar cell, and then your ganglion cell. It's what I'm gonna zoom in on. And for simplicity, I'm just gonna draw rectangles. So here is your photoreceptor, bipolar cell, ganglion cell. This is literally how I'm gonna draw it. I will draw the surrounding cells and the horizontal cells a little bit later, but here's basically how it works. We have the G protein right here. This G protein, I'm going to zoom in on. This guy, again for simplicity, I'm just going to draw a box. This guy has what we call 11 cis, oh my gosh, what is it called? 11, this is so embarrassing. I'm literally so embarrassed right now. I'm not, it's really not that big of a deal. Retinol, there it is. 11 cis retinol. And so what that means is that there is a cis bond on the 11th carbon of this retinol. And so this makes a little kink. And so when light comes in, sun, and hits this guy, this bond is broken, and then it straightens out, and then leaves. So what this does is it activates PDE, or phosphodiesterase. What phosphodiesterase is going to do is you have sodium get, I guess it is just channels, because I think a couple things both of them, but I'm just going to call them sodium. These sodium channels are activated by something we call CGMP. There it is. So this guy is what's opening this channel. So while this is activated, while the CGMP is activated, there is going to be an action potential in this cell, and glutamate is going to be released here. That's not necessarily what we want, but it's not necessarily bad. It's just when more glutamate is released here, there's less seeing. So in the dark, that's what's happening. However, when light hits this retinol and this activates the phosphodiesterase, this guy is going to kick this guy off, which closes this channel. And so this channel cannot depolarize the cell. So then what the depolarizing of the cell actually does is it opens up these calcium channels, which allow snare proteins to grab onto these vesicles, and then that's what releases glutamate. However, if we do not activate this guy, then this guy stays on, this channel remains open, depolarization still happens, and then glutamate is released. But we are inhibiting GDP, which kicks off this guy. No, we are activating PDE, which then kicks off the CGMP, which then decreases glutamate right here. Yeah, I gotta fix my chalk pen. Oh, please. All right. So you might be asking yourself, but why is a decrease in glutamate making it so I can't see? Let me tell you. What this receptor is this is an mGlu-R6 receptor. This guy is also stimulated by glutamate. However, this guy inhibits this channel, which is a sodium, another calcium channel. So if this guy is on, this guy is off. So this guy cannot bring in any cations, and so it can't depolarize this cell anymore, which means no glutamate is released from this cell, which is bad because on your ganglion cell, we have an AMPA receptor, which is also stimulated by glutamate. So if there's no glutamate, or I guess lesser glutamate being released here, then no action potential is going to go to the brain. So if we have a decrease of glutamate here, then this guy is inhibited. This mGlu-R6 receptor is going to be inhibited, which then opens up this, which allows potassium or sodium to come in, which then depolarizes the cell. These guys release glutamate, which then activates AMPA, which then you get a action potential to the brain. Woohoo! So light is, okay, there's an important rule I need to mention. Light always equals less glutamate. This is going to be a very important concept when I talk about the off-center and the surrounding cells, but this is what we call an on-center photoreceptor is. This is the process, okay? However, if this were an off-center, the only thing that changes is that this mGluR6 turns into AMPA, which then turns this guy on. So yeah, that's what that does. All right, let me talk about the little, what is it called? What is it called when we talk about the circles? One moment, please, if you could be so kind. What is this called? Receptor fields. That's what they're called. Okay. Receptor fields. When I talk about this receptor field, I'm only going to talk about on-center, off-center. If you're ever given an off-center on, wow. Okay, pause. That was so wrong. I'm going to be talking about on-center, off-surrounding, okay? If I start talking about or mention off-center, on-surrounding, just flip everything I'm saying. That simple. Super, super nice. All right, so if you recall from 5 seconds ago, this is a photoreceptor, photoreceptor, photoreceptor. This is also going to be referenced as this. This is on, I'm gonna use that as on center. This is a bipolar cell, ganglion cell, right? So light hits this guy. There's less glutamate here. This mGlu6, mGluR6, sorry. R6 receptor is inhibited, which allows action potential to happen here, which then increases glutamate here, and then action potential can go, right? So when we're talking about receptor fields though, we need to take into account this guy and this guy. So for simplicity, which is what everyone loves the most, is how I'm going to reference this is this guy gives 60%, this guy gives 20%, this guy gives 20%, or I might say something like this center photoreceptor has the potential to give 10 action potentials. However, the activity of these side guys is going to inhibit what happens to the center guy. But how does that happen? Let me tell you. So what we have right here are called horizontal cells because they look horizontal, but for now I'm drawing it as a square. So this guy, light hits it, same as the center photoreceptor. There's going to be less glutamate here, and right here is an AMPA. So then it causes this guy to release what we call GABA, which inhibits this guy because it increases, I think it increases conductance or opens a chloride and a potassium channel, which then depolarizes this cell, which causes more glutamate right here. But the actions of the center photoreceptor is greater than the actions of the side photoreceptor. So even though these two guys are both inhibiting the actions of the center photoreceptor, it's still gonna cause an action potential, okay? You with me? Perfect. So if light is over this whole thing, like the action potential is given is gonna be 6, right? If light is only given here, then this guy is helping, this guy's helping, this guy's not. You'd have 8 action potentials, right? If only light were right here, this guy off, this guy off, this guy helping. So there's gonna be 2 action potentials. So just depending on, this is off, over here, this is on, this is So depending on if we're talking about the on-center, off-surrounding, or the off-center, on-surrounding, and then where the light hits is going to be dependent on how well you see something. But this is basically how you see really good, okay? High resonance imaging, if you will. Anyway, that's how vision works. I don't know if I should be doing these videos in the context of I'm teaching someone who's never heard this before, or in the context of I'm explaining it to two people who are experts in this field, but I think I'd say the same either way. So. All right, lastly, let's talk about hearing, which I think I know the least about. That's okay. This is an ear, cute, right? Canal, tympanic membrane, think, yes. Or your eardrum. This is connected to 1, 2, 3 bones, and then this guy is connected to something that looks like that, right? Ha, ta-da! Actually, I hated how I drew that. These old. Oh my gosh, how am I doing this? Hold on. Okay. Yes, I like this a lot better. There we go. I don't know why that messed with my mind, but it did. All right, so this is what we call your external ear. I'm just gonna use E for short. This is just your canal, and so like sound comes into your ear, and then the vibrations come and hit your eardrum. So these bones are what we call, I think it's called middle ear, mid ear. I don't really know, but they're called the ossicle muscles. So we have the malleus, the incus, and then the stapes. So the function of these two guys together is that vibrations come and hit the eardrum, right? This guy is going to move this muscle, which then is going to move, or this bone, which is then going to move this bone, which is then going to move this bone, and it's going to hit what we call the oval window. This is air-filled. Air. So this amplifies the vibrations that we're receiving from outside, right? Because we don't want to lose any sound from when it's coming into the ear canal from when it's going to the cochlea. So this guy's going to amplify sound. And it does it in a couple of ways. One, it uses leverage. So these guys are just like long and skinny little sticks. And so they just, when they vibrate and hit each other, it basically amplifies. I don't know how I can better explain that, but that's what happens. I get it, but I don't know how to explain it. Another way it amplifies sound is that, like, if we were to compare the sizes of the eardrum versus the oval window, it's like the eardrum is like this big, so it's receiving like this much information. And then the oval window is like this big. So it's receiving the same amount of information, but in a smaller, more condensed space, which is going to help these vibrations move through the fluid of the cochlea. So the vibration of sound moves through these ossicle bones, and then hits the oval window. The oval window is then going to send vibrations going all over the place in this little guy. That's important because this is what we call the basilar. I am going to go grab another color. One moment, please. It's right over here. All right. This is what we call the basilar membrane. Right? And then, like we explained in class, I'm going to roll this guy out, and then I'm going to flip it this way. So then we're looking at the top of it, okay? It starts out short, cute, adorable. She goes, cha, cha, cha. So these guys are like short and rigid. These guys are long and wiggly. So the point of this guy is to allow us to hear really high frequencies, like I think the numbers were, what, 20,000 and like 20 hertz, was it? So depending on the frequency of the sound you're receiving from like your external environment or whatever, it's gonna hit in different, I'm getting chalk all over myself, this is funny. It's gonna hit in different parts of the basilar membrane, but that's chill. We want that to happen. However, as you age, these guys can start to deteriorate and like go away. Because of overuse, I mean, everything has to pass through it, so you start to not be able to hear as high of sounds, but I mean, you can still hear, like voices are at like 30, so like you're fine. There are before I forget, there are 3 different types of hearing loss. I actually cannot remember what they're called. Let me see if I can remember all 3 of them. But there's one where like your actual brain is damaged. I think that one happens from like strokes. There's one where there's something wrong with your external ear, and then one, so this could be like the calcification of these bones, or like a damaged eardrum, or like a clogged ear or something. And then there's also the last one is damage to your hair cells, and like this way. Ta-da. Okay, so now I'm going to erase this guy, but keep that in mind. I'm gonna keep that up. But I'm gonna take a cross section, and let's look into said cross section. Circle, smaller wiggly circle. This is what we call the tectoral membrane. I remember it because it sounds like tectonic plate, and tectonic plates move. All right, there's a membrane here, and then there's a membrane. I don't remember where I drew this next one, but there's a membrane. Let's just call it here. So this top tube, it's all one tube, but it folds on itself. So for now, I'm just gonna say the border is there. The top one is called, I forget, so I'm gonna look at my notes, my bad. It's called your scala vestibuli. Scala vestibuli. I'm gonna abbreviate as SV, and then this one is called your scala, I almost had it, tympani? All right, ST. So here's your SV, ST, right? I'm just trying to orient myself here, and you, but mainly myself, because I don't really know what's going on. But that is what we're looking at. And then right here in orange is the basilar membrane, right? So how the basilar membrane transmits sound to your brain, because like you get the vibration, it comes through your ear canal, it goes through the ossicles, and it comes through your cochlea, but how is it going to transmit that information is this next step, okay? So this guy is what we call hair cells. I should have done this bigger so you can see it, but you know what I'm talking about. All right, there is the outer layer, the inner layer. So these guys, I'm gonna zoom in on him. So here's the I don't want to call it tectonic membrane because that would just be embarrassing. Tectoral membrane. And then here is your, we'll call this basilar membrane. And then these hair follicles are like, not hair follicles, hair cells are right here, and their cilia are like all up in there. So when the tectoral membrane moves, vibrates, does its thing, these little hair cells are going to be moved, like squished. The little hair guys are gonna be like folded over to one side or the other. And on these little hair guys are mechanically gated potassium channels, okay? So when these guys bend this way, because this guy is squishing down, then this channel is open, and potassium can run into here, okay? Perfect. Keep that in mind. Next. This is filled with what we call perilymph. Same with this guy, but I don't want to write that again. And this is filled with what we call endolymph. Endolymph. I'm going to abbreviate because that took way too long to write. This guy is full, has a very large gradient, very large concentration, that is, of sodium. And then this guy has a very large concentration of potassium because we have these little guys. They were like rectangles in my notes, but I like lines better. These guys have, they both have a sodium-potassium component. Sodium-potassium pump, great. And then they both have 2 types of leak channels on them. They have potassium, which is moving it towards the cochlea, and then they have chloride, which is moving the chloride away from the cochlea. Okay, so these are like 3D. Pretend they're 3D. So sodium and potassium move in, potassium moves on, chloride moves out. Sodium and potassium move in, potassium moves on, chloride moves back away. Right, so that's where all of this potassium is coming from. And so, when, back to this other drawing, hope you can see it, when these mechanically gated potassium channels open up, a bunch of potassium is like, yo, I want to be in there. And so this is all just surrounded by this endolymph that just has tons and tons and tons of potassium in it, which is perfect because that's what we need. Because I can't remember exactly where the numbers come from. I guess I could look at my notes, bro. Guess who just didn't write it? Anyway, this membrane right here, because there's a bunch of sodium on this side and a bunch of potassium on this side, it almost makes this like a resting membrane potential, or just like a membrane potential, but it's not the membrane of a cell, it's like actual membrane of the body. But that just means that the driving force is like 120 millivolts. AKA potassium really wants to go in those little hair cells. And so this is attached to a nerve. And so then this adds positive charges to this guy, which then creates an active potential, which then transmits that information from the vibrations to your brain, and you hear things. And that's how that works. So, the end. That was really fun. I actually really like this unit, and I feel like I understood it a lot. Didn't do great on the test, but that was really interesting. Thank you for watching. I feel like I should end with something cooler. Wish I could do a backflip, but I can't. Anyway, that was fun. BRB.